In the photosynthesis, as I told you in the previous video, okay, for the photosynthesis we have light reaction and we have dark reaction. So for this video slide, I would be explaining to you, okay, regarding to the light reaction. Okay, so for the light reaction is when the light of energy they are converted to the potential chemical energy, okay, in the form of ADP and NADPH. Actually, in the simple sentence, okay, light reaction is the process of light of energy is converted to the ATP and NADPH. So in this reaction, they produce ATP and NADPH. Okay, in your respiration, it produces ATP and NADPH. But different in their photosynthesis, they produce NADPH because photosynthesis they are using a photo, a light. So because of that, they have a photo, okay, in the NAD and plus with photo P and H. Okay, so in the light reaction, they have two pathways, okay, which is we call first as cyclic pathway, second one is non-cyclic pathway. Okay, so for these both pathway, they produce ATP, okay, from the ADP plus PI. Okay, so as you know before, in order we generate the ATP or we produce the ATP, they should be from the ADP plus PI. Okay, so similar in this photosynthesis process. So what the name of that process? Okay, we call as photophosphorylation. Okay, remember in your respiration, when you produce the ATP, we call that process as substrate level phosphorylation, or we call as oxidative phosphorylation. Okay, so they have the phosphorylation there, which is transfer of phosphate to the ADP and become the ATP. Okay, because photosynthesis they are using a light, so because of that they have the name photophosphorylation. Alright, so before we go deeply or detail regarding to the step in the light reaction, so you need to know about the basic information, okay, before we learn more about the light reaction. Okay, in the light reaction, they have the substance or the protein that we call as photosystem. Okay, this photosystem, they are surrounded with the light harvesting complexes. Okay, in the center they have the reaction center. Okay, you may look after this the illustration of the photosystem. Okay, regarding to where they have the light harvesting complex and where they have the reaction center. Alright, so each light harvesting complex that consists of the pigment molecules. Means in the harvesting complexes they have several pigments. Okay, the primary pigments and all the accessory pigments that can found in the leaf. Okay, and for the reaction center is a protein complex, okay, that have a chlorophyll A, which is two chlorophyll A, okay, and a molecule that we call as primary electron acceptor. Okay, this is the photosystem for the plant. Okay, as I mentioned before, photosystem is including light harvesting complexes, okay, which is the green color. You can see the green color is all the accessories pigment. Okay, and they have the reaction center in the middle of the photosystem. Okay, in the reaction center they have two green color, which is a chlorophyll A. Okay, and they have the primary electron acceptor. So how this photosystem harvests the light? Okay, because the photosystem actually is the first substance that absorb the light or capture the light. So it happens, okay, when a photon means the energy of the light, okay, they are strike a pigment molecules, okay, in a light harvesting complexes. So the energy when the accessory pigment, which is light harvesting complex pigment as the accessory pigment, absorb the energy, that energy will be passed from one accessory to another accessory molecule or accessory pigment until they reach to the reaction center. So at the reaction center, first they reach at the chlorophyll A. Okay, And when chlorophyll A, they accept or they receive the electron, the energy from the accessory pigment. So the electron from the chlorophyll A will be excited, okay, to the primary electron acceptor. So because of this primary pigment, which is chlorophyll A, we call they have their uniqueness or the spatial. Spatial is because of their molecular environment. Okay, how about their environment that you can see? Is regarding they have the best location, which is they at the middle area, which is they are associated also with all the accessory pigment that able to trap the light and channel it to the chlorophyll. Okay, so for the photosystems, they have photosystem 1 and they have for the photosystem 2. So for this photosystem, as I mentioned before, they also as a pigment. So the different pigment, they able to absorb different wavelengths. So same as photosystem 1 and also photosystem 2, which is okay, for the chlorophyll, okay, at the reaction center of the photosystem 1, okay, we call as P700, which is why we call as P700 for the PS1, because the best or the most efficiently absorbed light at the wavelength 700 nanometer. Okay, different to the photosystem 2, which is their chlorophyll, okay, at the reaction center of the photosystem 2, okay, we not call as P700, but we call as a P680. Because why? Because this pigment is best, okay, or most efficiently absorbed wavelength, okay, at 680 nanometer. Okay, that is the first difference, okay, between the photosystem 1 and 2. Okay, and second difference is okay, who of the phototrope using photosystem 1 or photosystem 2 and photosystem 1. Okay, as you can see for photosystem 1, it might be used by the prokaryote. Okay, as I mentioned before in your first slide regarding to who actually do the photosynthesis. Not only the green plant, but also involve the algae, okay, and also the prokaryote, which is we call as cyanobacteria. Okay, different to the photosystem 2. Okay, you can see prokaryote require, okay, photosystem 2 and photosystem 1. Means for the green plant and also the algae, when they do the light reaction, they require both, which is they require photosystem 2 and also photosystem 1. Okay, now we move to the most complex step. Not complex actually, just a simple if you understand about this step. So for the light reaction, they have two pathways. Okay, which is first, I told you cyclic and non-cyclic. So we look first on non-cyclic electron flow or non-cyclic pathway. So for the non-cyclic pathway, okay, you have 1 until 8 step that you need to remember. Okay, which is if you look on this, so explain you in the overall way. Okay, which is number 1, you can see from the light means here, okay, the accessory pigment have been absorbed the photon of energy, light energy, and it will be transfer or channel it to another accessory pigment until reach to the chlorophyll A. Okay, that is number 1. So when reach to the chlorophyll A, P680, so the electron at one of the chlorophyll A will be excited to the primary electron acceptor, which is step 2. Okay, then step 3, you can see that they have the water. Remember, for the light reaction, they require the water. So because of that, at step 3, okay, the water, okay, through the process of photolysis, it might be using light, okay, break down the water, okay, produce hydrogen ion, produce oxygen, and produce the electron, 2 electron. So this electron from the water will be replace the electron that are lost to the primary electron acceptor. Okay, that is step number 3. Okay, step number 4. Okay, just now, when the electron first from the chlorophyll A excited to the primary electron acceptor, it will be go down to the electron transport chain, which is the electron will be passed through the plastoquinone first, PQ. Okay, then electron will be passed to the cytochrome complex. Okay, remember about the electron transport chain in the respiration process, in the aerobic respiration, which is that is about the electron will be passed to the one multi-complex to another one multi-complex through the process of redox reaction. Means one will be oxidized and then reduced and oxidized and be able to pass the electron. Okay, similar to step number 4, which is the electron would be passed first to the plastoquinone PQ. Okay, this PQ will be oxidized. So when they are oxidized, cytochrome complex will be reduced. So they accept the electron from the PQ. And the electron from the cytochrome complex will be oxidized. Then when they are oxidized, the electron pass to the PC, plastocyanine. So plastocyanine will be reduced. Okay. And when the electron pass through the electron transport chain, step number 5, it will be produced the ATP. So first product in the light reaction is ATP. Okay, so number 5. Okay, then you look on the step number 6, which is step number 6 is regarding what happened to the photosystem 1. Okay, the process is similar to the step number 1 in the photosystem 2, which is at step number 6, okay, light energy have been absorbed by the accessory pigment and it passed from the accessory pigment to another accessory pigment until reach to the P700, means at the chlorophyll A P700. So what happened to the electron at the P700? It will be excited to the primary electron acceptor. So that is step number 6. So when step number 6, okay, after that step number 7. Okay, before step number 7, you can see the electron, okay, from the from the photosystem 2 just now when they are going down to the electron transport chain, this electron will be fulfill the hole at the P700. Okay. Then step number 7, the electron that are excited first from the P700 will be passed to next electron transport chain. But this electron transport chain, they're not using a similar protein, PQ, okay, cytochrome complex, and PC, which is the electron should be passed through the protein that we call as ferridoxine, FD. So DFD, the electron will be, okay, oxidized and pass the electron to the NADP plus, okay, by the enzyme that we call as NADP plus reductase, and NADP plus is reduced to the NADPH. So because of that, they produce second product, which is NADPH. Okay, if you cannot remember, okay, or digest what I'm saying you about the step number 1 until number 8, if you not understand my explanation, it's okay, because the next slide, okay, in this slide, you will be explained again, okay, in the sentences, okay, in the short AC, how actually the process of non-cyclic electron flow have been produced, the ATP and also the NADPH. Okay, so now here, okay, a photon of light, they strike a pigment molecules in a light harvesting complex, which is what happened in the PS2 just now, which is the accessory pigment absorb the light from the absorb the photon from the light, and it will be channel it until reach to one of two P680 chlorophyll A. And chlorophyll A electron will be excited to the reaction center. Okay, so one of the P680 electron excited to the higher energy state. Okay, and this electron is captured by the primary electron acceptor. Okay, at the PS2. Okay, then step number 3. It involves about the water, as I told you just now. So the process is hydrolysis process. Okay, which is, we call this process as photolysis. Okay, lysis split the water or lysis the water, okay, using the hydrolase enzyme. So split the water, okay, to the 2 hydrogen ion, okay, and 2 electron, and also the oxygen atom. Alright, and the electron that supply one by one to the P680, because it might be replacing an electron that lost, okay, to the primary electron acceptor. Okay, and the oxygen that they are formed from the splitting of water, okay, immediately will be combined with other oxygen atoms, okay, forming the oxygen. So from the oxygen atom, okay, so oxygen atom 2, okay, die, so they become the oxygen. Alright, and step number 4. Step number 4, where the photo-excited electron, means the electron that are excited from the chlorophyll A, okay, at the PS2, from the primary electron acceptor, it might be going down from the PS2 to the PS1, okay, through or via the electron transport chain. As we can see before, the transport chain, the electron carrier should be PQ, cytochrome complex, and plastocyanin. So using this electron carrier, they are able to pass the electron using the reaction. So then the process exergonic fall of the electron, okay, to the lower energy, because if you look on the illustration just now, it might be from the excited primary electron acceptor, it will be go down to PQ, cytochrome complex, and PC. So from higher energy level to the lower energy level. So because of that, it might be synthesis the ADP. Okay, and the next step is regarding what happened in the photosystem 1, which is at the photosystem 1, okay, all the light harvesting complexes, which is all the accessory pigment, absorb the light energy, and it excited the electron from the P700 to the primary electron acceptor. Okay, then when the electron at P700 is excited to the primary electron acceptor, so it creating the hole, okay, in the P700. So what function of that electron hole is to fill by the electron that reach at the bottom of the electron transport chain from the PS2. Because just now, okay, first we have the electron that are going down the PQ, cytochrome, and PC. So this electron will be replaced or will be filled the hole, okay, at the P700. Okay, and the photo-excited electron pass from the PS1 primary electron acceptor. So it will be going down second electron transport chain, okay, through the protein that we call as FT, which is ferridoxine. Okay, and the NADP plus reductase, which is the enzyme, okay, will be transfer the electron from the FD to NADP plus, and NADP plus will be reduced to the NADPH. So in the overall process of the non-cyclic, it will be produced ADP and NADPH. Alright, now we look on the cyclic electron flow. Okay, this one is also one of the light reaction. because light reaction, they have non-cyclic and they have the cyclic. So the cyclic reaction, they have a simple pathway, which is the pathway is always continue in the cycle shape. Okay, it's different to the non-cyclic, because non-cyclic, they involve two photosystem, which is from the photosystem 2 to the photosystem 1. However, for the cyclic electron flow, they only involve the photosystem 1. Okay, and the electron transport chain also have 1, okay, not 2 as the non-cyclic electron flow. So for the cyclic electron flow, the electron cycle, okay, back from the ferridoxine, okay, to the cytochrome complex, and then continue to P700 chlorophyll, okay, in the PS1 reaction center. So they not produce any NADPH and also not release any oxygen, because they not involve the photolysis process. They just only produce the ATP. Because why? Because the electron, when they are excited, they must be go down the electron transport chain. Okay, so they produce the ATP only. Okay, this one is the illustration of the cyclic electron flow. So you can see that they only involve the photosystem 1. Okay, where they have the light energy. Okay, and accessory pigment will be capture it and transfer from one accessory pigment to another accessory pigment until reach to the P700, which is until reach to the chlorophyll A. Okay, P700. So when the electron at P700 can receive the electron of chlorophyll A, P700, will be excited to the primary electron acceptor. That are similar or same process, okay, as non-cyclic pathway at the first stage. After that, okay, after they are excited, electron should be dropped to the ground state, which is from the primary electron acceptor. It might be through the redox reaction, pass through the electron transport chain, which is ferridoxine, cytochrome complex, and pass to the PC. Then they produce the ATP. So this electron from PC will be received back at the P700. So because of that, the process is always in a cycle process. Okay, so now we look on the photolysis, because photolysis is involved in the light reaction. So means what we can say that for the photolysis, there are the parts of the light dependent reaction, which is for the non-cyclic reaction, okay, not for the cyclic. So this photolysis, they refer to the use of the light energy in order to break the water molecules and to obtain the electron for photosynthesis. Why we call the obtain the electron for photosynthesis for the lysis process? Because you need to remember for step number 3, okay, which is when they split the water, okay, the electron from the hydrolysis of water will be replace the electron that are lost to the primary electron that are lost at the chlorophyll A, P700 to the primary electron acceptor. So because of that, function of the photolysis, okay, is to replace the electron that pass by the photosystem to the electron transport chain. And in the different terms that we can say that the water is the electron donor, because they donate the electron from the splitting of the water to the hydrogen ion, electron, and also the oxygen. So this process also is the process how the oxygen return to our atmosphere. Okay, now we look on the relationship between the light reaction and the chemiosmosis. Okay, why chemiosmosis involved in the light reaction? Because light reaction, they produce the ATP. Remember, for the chemiosmosis, is about the electron pass from the one carrier to another carrier. Okay, in the redox reaction, means in one protein will be oxidized and one protein will be donate the electron, and another one will be receive the electron and become reduced. Okay, so when they pass the electron from 2 to 3, so 2 will be oxidized and number 3 will be reduced. Okay, that is the electron transport chain. So hydrogen ion that removed from the stroma, okay, are deposited in the thylakoid space. So this phenomena or this environment will be storing the energy, okay, as a proton motive force, or we call as hydrogen gradient. Remember, for the chemiosmosis, they require different concentration of hydrogen ion. So we call this energy as proton motive force, or also as the hydrogen ion gradient. Okay, so for this one, three steps actually that are contribute to the proton gradient, okay, which is when the water split, okay, by the photosystem 2, because for the non-cyclic, okay, the water split, okay, by the photosystem 2, not photosystem 1. So when they are split, so they will contribute or they will increase the number of the hydrogen ion in the thylakoid space. Okay. Number 2 is regarding to when the electron pass from the PQ, okay, to the cytochrome complex. So the hydrogen will be pumped, okay, from the you can see after this we have the illustration. So from the stroma, it will be passed or across or translocate across the membrane, okay, into the thylakoid space. Okay, number 3 is regarding to remember, NADP plus will be reduced to NADPH. This process occur in the stroma. So because of that, hydrogen ion also will be decreased in stroma, because hydrogen ion is removed, okay, by taken up by the NADP plus. So for the hydrogen ion that are pumped from the stroma and enter the thylakoid space, these will be produce different hydrogen ion concentration. So when they have different hydrogen ion concentration, it will be power the ATP synthase, which is enzyme to synthesize the ATP to produce the ATP. Okay. So because of that, for the light reaction, they produce ATP and also NADPH. Okay, this one is the illustration regarding to the chemiosmosis and light reaction. So you can see that photosystem, they are embedded in the membrane. Okay, what membrane is the thylakoid membrane? So you can see the process for the water is hydrolyzed, so increase the hydrogen ion in the thylakoid space. Okay, step number 2, just now, in order to contribute the different concentration of hydrogen ion, when the electron is passed from the plastoquinone to the PQ through the cytochrome complex, it will be passed or it will be pumped the hydrogen ion from the stroma and enter the thylakoid space. So this process 2, number 1 and number 2, will be increase more the concentration of hydrogen ion, okay, in the thylakoid space. And number 3, which is how an ADP plus, when they are reduced, so it will be accept the hydrogen and produce the NADPH. So this process will be increasing the factors that will be reduce the number of the hydrogen ion in the stroma. So because of that, in the stroma, they have low hydrogen ion concentration, and in the thylakoid space, they have high hydrogen ion concentration. These different hydrogen ion concentration will be makes the ADP synthase to produce the ATP from the ADP plus Pi. And these both products, which is ATP and NADPH, will be used by the Calvin cycle. Okay, and this one is regarding to differences, okay, between the non-cyclic and cyclic pathway, means in the light reaction. So you can make some differences using some using 1, 2, 3, 4, 5 different items or 5 different things that you can distinguish, okay, between non-cyclic and cyclic. So what you can say that for the first one is regarding to electron source. Okay, electron source for the non-cyclic should be from the hydrolysis or photolysis of water. Okay, however, for the cyclic, they have no electron source. So from the process of photolysis, it will be split to the hydrogen, to the electron, and to the oxygen. So because of that, the things number 2 is regarding to the oxygen release. So of course, in the non-cyclic, the release, but in the cyclic, it's not release any oxygen because they not involve any water. Okay, in the cyclic electron transport. Okay, as the terminal electron acceptor, also no for the or none for the cyclic because the process is cycle. They do not have terminal electron acceptor. But for the non-cyclic, okay, NADP plus as the terminal electron acceptor because NADP plus is the last that receive the hydrogen before they reduce to the NADPH. Okay, number 4 is regarding to energy. What the energy that they form? They form for the non-cyclic is ADP and NADPH. However, for the cyclic, they not produce NADPH but only produce ADP. And the last one regarding to the photosystem. Photosystem for the non-cyclic, they involve both PS1 and PS2. However, for the cyclic, they only involve the photosystem. Alright, and the last one is regarding to general distinguish or differences, okay, between photosynthesis and aerobic respiration. As we learned before about the aerobic respiration, okay, this process we call types of metabolic reaction is catabolism. Why catabolism? Because we break down, okay, raw material. We break down the glucose, okay, using the presence of the oxygen. So because of that, raw material should be glucose and also the oxygen. And we produce from the aerobic respiration CO2 and water. Okay. However, when you look on the photosynthesis, okay, there are different types of the metabolic because photosynthesis, they produce the glucose, which is the end product, and the raw material is only contra to the aerobic respiration, which is raw material of the photosynthesis is the CO2 and water. However, for the end product, they synthesis the glucose and oxygen. Okay, the site also different because for the photosynthesis, they occur in the chloroplast, and for the aerobic respiration, they occur in the cytosol and mitochondria. Because for the cytosol, they involve for the glycolysis. However, for the Krebs cycle, okay, for the what else? For the oxidative phosphorylation, which is electron transport chain, and also the chemiosmosis, they happen in the mitochondria. Okay, and you have the ATP production, okay, we call as photophosphorylation. For the aerobic respiration, we call as substrate level phosphorylation, and also the oxidative phosphorylation. And they have several things that you can make differences, okay, that you can difference between the photosynthesis and the electron and also the aerobic respiration. Okay, so the last one, if you can see that the terminal electron acceptor, okay, for the electron transport chain also you can make one difference regarding to photosynthesis and aerobic respiration, which is, okay, in the non-cyclic electron transport, okay, they have the NADP plus, okay, as the terminal electron acceptor, where this NADP plus can be reduced to NADPH. Okay, however, for the aerobic respiration, you can see the last one in the electron transport chain. Okay, the electron will be received by the oxygen because oxygen is more electronegativity. So because of that, terminal electron acceptor for the aerobic respiration is oxygen. Okay, that's all, student, for the aerobic respiration and also for the photosynthesis. And if you can't imagine the process, so here they have some simple process regarding to the raw material and produce the production, okay, of both photosynthesis and respiration. Okay, that's all. Thank you.